Hello everybody. So we're going to talk about inheritance and how genes are passed down from generation to generation. Now, if we start with just my family, right? So here's my dad. Let me get my laser pointer out. Here's my mom. This is one of my grandpas, and this is my other grandpa and my grandma, right? My grandpa now is actually the only one left of my grandparents, but he's 100 years old right now. So but I still have some of my family around, and when we, look at my dad, look at my mom, we can see that there are some traits that I share with them. And then when we look at my brother and I, this is a picture of a much younger me, and here's my brother Tom. And you can see that he and I share some characteristics, right? We have the same color hair. This is us taking a stupid picture after we had been skiing all day. But, our hairstyles aren't necessarily that similar. He's now bald, I'm not. But we have the same eye color. Our eyebrows are kind of similarly shaped. Our noses are similarly shaped. He's 6'1", I'm 5'8". So there are some differences here. But, we still look... we can certainly look like brothers, right? Now, when we then go to the next generation, like my children, right? We can see, here's my son Jordy, here's my daughter Laya. Again, they have the same hair color, they have the same eye color, skin tone is very similar, right? So, we know that genes are passed from generation to generation. And, some of my son's characteristics would be very similar to maybe my grandpa, right? Or even like my brother, right? We can see that there's family resemblance. Now, the old idea of inheritance was this thing, right? Where basically this was a sexist idea, but a lot of people thought that there were tiny little babies in sperm, and we know this is wrong though, right? Because when you think about it, everybody would be then just an exact copy of their dad, but they're not, right? Each baby is a mix of mom characteristics and dad characteristics. And we can see this with my son Jordy, okay? So if you look at these characteristics, these are just simple, very simple characteristics based on one gene. And we can see that I do not have a widow's peak. My wife Andrea does not have a widow's peak. Neither does Jordy. Mid-digital hair now, that is, look at your hand, and there's the bone that, look at your finger, and the knuckle that is with your fingernail, and then the next knuckle, basically like the next bone, right? That's your mid-digit. Look to see whether you have hair on that mid-digit. And both me, my wife, and my son have that. If you look at our earlobes, my son and I have attached earlobes, but my wife has a free earlobe. So it's like kind of like a free swinging earlobe as opposed to like that's like attached completely. If you look at my family's pinkies, take both your pinkies and stick them out and lay them next to each other so your pinkies are like touching, right, along the pinky. And then look at the tips of your pinkies and see if the tips of your pinkies either touch or kind of bend outwards. And mine are definitely bent, whereas my wife's and my son's are straight. Give yourself a thumbs up and look at your thumb. Does your thumb point backwards? If it does, it's a hitchhiker's thumb. And then, so all of these genes are these very simple, all these characteristics are by a single gene. They're like controlled by a single gene. And where we see a little bit difference is in hair and blood type, which we'll get to here soon. But you can see here's my blood type, here's my wife's blood type, and here's my son's blood type. So let's just look at this earlobe gene. Earlobe trait, I should say, right? So here's an attached earlobe, and my wife has this free earlobe where it's kind of like swinging kind of below the ear. Now, what a gene is a sequence of actual letters of DNA that codes for a trait, the trait being earlobe shape. Then an allele is the form of that gene, whether it's attached or free. Now, we are what are called diploid organisms, right? So my son Jordy has one earlobe gene from his mom, one earlobe gene from me. Now, free earlobe is dominant. Okay, what that means is a dominant allele masks the expression of a recessive allele. Okay, so if you have the gene for free earlobe, you're certainly going to show that gene or show that trait. So what we know is, since I have an attached earlobe, I'm going to draw this as little e, little e. I'm going to like make shorthand for letters, right? So what this little e is actually stands for is a sequence of maybe thousands of letters of DNA that are recessive. Okay, so this little e is my genotype, which shows me the actual alleles that I have. I have 2 copies of the attached earlobe gene, or I should say the attached earlobe allele on the earlobe shape gene. Then the phenotype is how those alleles are expressed, so what we actually see, and that is the attached earlobe. So if I have 2 little e's, what did I give my son Jordy? I had to give him a little e. Now, my wife, we know she has at least one big E, right? Because she has the free earlobe, which is dominant. So we know she has one big E. Now, this big E is referring to the dominant allele. So if someone were to be big E, little e, they could still be this free earlobe. Or they could be big E, big E. Now, we don't essentially, at first, if we were to just look at my wife, we don't know whether she would be big E, or big E, little e, because since the big E is dominant, since the free earlobe is gene allele is dominant, it could mask what she has. However, when we look at it, since my son Jordy has we know that for him to show the recessive trait, he had to have gotten another little e. So then we can backtrack back up to my wife, and we know that she was little e. I'm going to skip this actually. Okay. So what we call this then is homozygous. Homo meaning same. So it shows that I am homozygous recessive. My son Jordy is also homozygous recessive. And my wife then is what we call heterozygous. Hetero meaning different, right? So she's got different genes here, or different alleles, I should say. So we can think of this a little bit more complicated, okay? If Andrea, my wife, is homozygous dominant, what percentage of our babies will have free earlobes? So think about this for a second. Oops, sorry. And what we can do this with is called a Punnett square. Is the pen going to work? I'm having some problems here. Let me see if I can do it this way. Nope, that's not going to work. I'm sorry for these technical difficulties. So I'll have to just do it with words, and I'll come back to this in class when we meet on Wednesday. So if my wife is homozygous dominant, that would mean she could only give a big E, right? And I would remember I'm homozygous recessive, so all of our babies would have free earlobes. But if she's heterozygous, then 50% would come up with free earlobes and 50% would be attached. So this is all having to do with one gene. What I want to do is looking at a little bit more complicated modes of inheritance, we call them. So, so far we've looked at one gene with only 2 alleles. But there's some genes that have we're still working with one gene here, but have more than one allele. So, this is incomplete dominance, where you have a big R and a little r. And when you're homozygous red, the flowers are red. When you're homozygous recessive, flowers are white. So homozygous white, the flowers are white. But when you're heterozygous, then you get a pink flower. What we see here is heterozygotes have a phenotype that consists of the mix of both alleles. So a pink is, the mix of half red and half white here. So it makes things a little bit more complicated, but you can still do these like Punnett square type things and make it pretty easy to figure out what the alleles or what the phenotypes will be. Where it starts to get a little bit more tricky then is where you get like lots of alleles with one gene and this thing called codominance, okay? And what we actually see is most genes have more than 2 alleles. Think about eye color, right? How many different eye colors are there? Well, there's brown, there's blue, there's green, there's hazel, there's kind of yellowish, right? And people that are kind of in between, right? So there's lots of different genes, lots of the different alleles for the eye color gene. Now, in this specific thing called codominance, it's a interesting way in which we have A, B, and O blood type, okay? And the A and the B are codominant in the sense that they're both expressed equally. An example here is, like, take this guy's shirt. Horizontal stripes, vertical stripes. Now, if they give both those genes to the kid has horizontal and vertical stripes. So, for blood type, what we see is A and B are dominant, like codominant with each other. So that when you have AA, or A, and this little I is actually standing really, I would normally write that as an O, but the I is technically right. But if you have a genotype that is AA or AO, you get blood type A. If you have BB or BO, then you have blood type B. If you have an A allele and a B allele, then you have blood type AB, represented here by the circles and the diamonds. And then OO here, or in this case little I, which gives you blood type O, which means you have none of these carbohydrates on the outside of the red blood cell. So when you do Punnett squares, whenever you see AB, you absolutely know what genotype they have because their genotype has to be AB, right? These are the phenotypes. When you see a phenotype of blood type O, you know what they are. However, when you see phenotypes of blood type A, we're not really sure whether it's AA or AO. With B, it could be BB or BO. So this gets a little bit confusing, and when we come back from class, I'll do some Punnett squares up on the board to try to make this make sense. So we're going to come, when we come back on Wednesday, we're also going to do a worksheet. So be ready in class to do a worksheet where we're going to basically figure out some things about my family. And using this sheet, we can do some things and should be a good time. And we'll learn a little bit about how to do Punnett squares and how to do some, like back and forth looking at how genes work together to create the traits that we see. So one other thing we can do, though, is pedigrees. And you've done these with lab a little bit, where we're looking at Hutchinson's disease. And the best way to figure out like what genes are in your family are to do what's called test crosses. Okay. Let me go back here real quick. Let's say we were in a position here where my son didn't exist, right? And we were trying to figure out what the genotype of my wife was. Okay? Now, if we were to have a kid and they came up with a free earlobe like this, so if Jordy had a free earlobe, what does that say about this blank letter? And if you think about it, the answer is actually nothing, right? What happens if I have 2 kids that have a free earlobe? 3 kids, 4 kids. Once we start getting high enough, lots and lots of kids with all of them having free earlobes, we can with some confidence say that there is, big E here. If all of my kids were to have a free earlobe. But it's still possible to flip a coin 5 times in a row, 10 times in a row to get heads. And that's what's essentially happening here, or could be happening here, if I were having kids with free earlobes and we didn't know what this other letter was. So when we go to, But one way we can figure out a little bit potentially about people's genes is by using pedigrees, okay? And what we can do is compare the probabilities to actual people, and we can figure out that whether people have dominant or recessive traits, and whether certain people are heterozygous, because in this case it makes a difference. So this is with a deafness allele, right? And this Jonathan guy is he's deaf. He has 2 recessive alleles here, and it has a deafness gene. So we can then go back up to his parents and know that Joshua and Abigail were heterozygous because they weren't deaf, but then their kid was. So we can go back and feel that. But we don't know what Abigail is, right? We know she has at least 1 big D, but did she get, who did she get that big D from? We don't know. And, that could be heterozygous hiding in here. And then we know, since Jonathan had some kids that were deaf, this and this, we know that then Elizabeth was heterozygous also, right? So you can kind of go back and forth on this pedigree to figure out different things about this family tree. Now, when we talk about pedigrees, it's oftentimes in the sense of looking for diseases or human genetic disorders. And what we see is most disorders are recessive because most disorders are bad, right? And when we are looking at all the different disorders, what we see is that they're not evenly distributed across ethnic lines. Okay, so Europeans and European Americans, cystic fibrosis is basically very common with Caucasian and white people, right? Whereas something like sickle cell disease is much more common in African populations. And sickle cell disease is one that's actually really interesting because it creates a resistance to malaria. Normal red blood cells are these like donut shapes, but sickle cell disease people have these like kind of half moon shaped, sickle cell shaped red blood cells. They can get trapped and actually cause like strokes and blockages. But the fact that they have the sickle cells, you can see the sickle cells right here, makes them be essentially immune to malaria. So when you look at a map looking at like prevalence of this is malaria in green, we can see a lot of that really overlaps with the sickle cell trait, because basically when these waves of malaria would come through and kill a bunch of people, the people with sickle cell in this equatorial Africa, somewhere around 12% of them have sickle cell disease, and that's really, really high for actually a deadly disease. So when we look at this human disorder chart, what we see is basically there's like inbreeding on like continental scales that has caused some certain types of diseases to be prevalent in certain populations, and that's what makes it really quite interesting to track within families and where potentially you can get some of your genes. So what we do see though is dominant disorders are really rare because they're heavily selected against, right? If a disorder is assured to come up and never be able to hide in a heterozygote, it's going to show up more. The phenotype of the disorder will show up more. But what we got to realize is that because a trait is dominant doesn't mean it's really common, doesn't imply prevalence. Okay, and what we see, things like certain types of little people, they often are the heterozygotes are the only ones that actually create viable offspring. So achondroplasia, which this little person here has achondroplasia, and we're not really sure whether their kid does or not. In this picture, I can't tell at least. But an achondroplasic adult is heterozygote, so they could have adult children or like normal sized babies, and so all of the little people basically are carriers for this disorder of achondroplasia. All right. I want to then talk about one more type of inheritance, okay? And this is called polygenic inheritance, where poly means many, genic means genes. And what we see for things like skin color or height or there's plenty of genes that like muscle size can be considered polygenic. Basically what they are is there's like a bunch of different genes that have different alleles that all work together to create the final phenotype. So an example here can be like skin color, okay? What we have here is 3 genes, the A, B, and C gene, and all have dominant and recessive alleles. Now this is a simplification of skin color, but what we see is when you have all recessive, that means basically your body doesn't produce that much melanin, and you would have very, very light skin. Okay, whereas if you were to have all dominant, that means you'd be like producing a bunch of melanin, you would have very dark skin. Okay, and then there's all sorts of these gradations in between based on the number of dominant alleles that you can have. And you can create 1, 2, 3, 4, 5, 6, 7 skin colors basically just with 3 genes. Now reality is a little bit more complicated, and there's many genes that can affect it. So if you can think of just with 3 genes, you create these 7 different skin colors. Add another gene, and all of a sudden you got a whole lot more. The thing is, these traits are often masked by environmental variation, right? So you might be somewhere here, maybe I'm somewhere here, I should say, for my skin color, right? But my skin color highly varies. In the winter I'm super white. In the summer I can actually tan pretty well. So, it can vary over time. Even, like someone who has the exact same genotype as me, so let's say like my brother, right? If he works indoors more than I do, he might not have as dark a skin as me at a specific time. So, polygenic inheritance, once we start talking about that, is much more complicated than things like we started talking about with earlobe shape or whether your pinkies are straight or not, right? Those are single gene inheritance that are very easy to spot and figure out quite quickly, but polygenic inheritance makes it much, much harder to work with. All right, well, that's it for today. I'll see you all on Wednesday.